Hi, my name is Elisa Kaneko, and I am a professor of biology at Gannon University in Erie, Pennsylvania. Ask your parents or elderly relatives about aging, and they will tell you that getting older is hard. As we age, our joints stiffen and our muscles weaken. Diseases such as arthritis can speed up the natural aging process and make everyday tasks like buttoning our clothes or walking upstairs much more difficult. Today I will talk to you about 3 topics: 2 diseases affecting the joints of our hands and our legs, a protein in our bodies called relaxin, and the different ways we study relaxin in my lab. At the end of this talk, you will understand how a protein called relaxin may prove to be a non-surgical treatment for arthritis and other diseases, and how you can play a role in studying this protein. Relaxin is found in both men and women, but pregnant women make the most relaxin. Relaxin's claim to fame is its role in the last trimester of the pregnancy. It softens and slightly stretches the ligaments and other tissues in the hip bone so that they can spread apart slightly as the baby moves through the birth canal. Relaxin also brings about other changes in pregnant women, like wider feet and an excessive arch to the lower back. In women who are not pregnant and in men, relaxin is still made, but in much smaller amounts, so its effects are less dramatic. Its main role for most of us is to break down collagen, another protein of the body. Collagen is found in the ligaments and tendons of our joints and in tissue found under the skin of our palms, known as fascia. This fascia connects the skin to the underlying bones and muscles so that they act as gripper surfaces. I work with local physicians and scientists. We want to use relaxin to help treat diseases like arthritis, which stiffens the ligaments in the joints of the body. Surgeons also have to go in and cut out that stiff and thickened ligament, freeing the joints to move. Surgeries are very expensive procedures. Not only do you have to pay the surgeon, the nurses, and the hospital, you also have to take days off from work, and your normal activities are disrupted. In the future, it might be possible to inject relaxin into the joints to keep the ligaments from stiffening. This saves time and energy for everyone. Another disease is called Dupuytren's contracture. It was named by a French physician in the 17th century. Dr. Dupuytren described a condition in which the fascia of the hands becomes contractile, like a rubber band. As this tissue contracts, it pulls the fingers into the palm, making a fist. Eventually, the patient cannot use the hand at all. For centuries, the only treatment has been to cut out the rubber band-like parts of the palm, freeing the fingers and allowing full use of the hand. In the future, relaxin could be injected into the thickened parts of the palmar fascia, softening the collagen and freeing up the fingers from their contracted state without surgery. Again, this will save patients time, money, and pain. They won't miss work and they won't disrupt their normal activities. Our research looks at how relaxin interacts with the cells making collagen in ligaments and fascia. These cells are called fibroblasts. Remember, relaxin and collagen interact with one another. Now, relaxin's effect is not as straightforward as it could be. It seems to act in what is described as a signaling cascade. This cascade is like a game of telephone in which one person tells a message to a second person, who in turn tells a third person, who in turn tells a fourth person, until the message is spread to everyone in the room. Relaxin in this cascade binds to one protein on the surface of a cell, protein A. Protein A in turn binds to protein B, which interacts with protein C, which affects protein D, etc., etc. Eventually, at the end of this cascade, the collagen protein is broken down and the tissue softens. To look at the interactions in this cascade, we use several different types of experiments in my lab. One of them involves microscopy. You used microscopy in the freshman lab when you were studying bacteria and the different cells of the body. To look at fibroblasts and other cells found in the skin, we grow cells in plastic dishes and we tag them with proteins that glow in the dark. Now, we need to go back to grade school when you went on a field trip to the Natural Science Museum. You might remember seeing a lot of rocks on shelves in natural light. You flip a light switch and you turned on a black light, and all of a sudden the rocks glowed yellow and red and green. Small molecules on those rocks made them glow in the dark. In my lab, we used that black light and those same small molecules to make the cells glow in the dark. We attached the small molecules to several different proteins in the cells so that each protein is tagged with a different color. When you look in the microscope, different parts of the cell glow yellow, red, and green. This technique is called indirect immunofluorescent microscopy. It's indirect because we're looking at the glow in the dark small molecules instead of the actual proteins like collagen. Immuno refers to antibodies. Antibodies are proteins our bodies make when we are infected with a virus or a bacteria. And these antibodies are a kind of connector or bridge between the collagen and the glow-in-the-dark molecule. We call this technique fluorescent because we use black light to view the cells. In the rest of this talk, I will talk about other techniques used in my lab, some of the results we found using those techniques, and how you can take part in this research. Let's get started.